My hand looks empty, but in real life, thousands of organisms live there. They are small and invisible. They lived there long before the dinosaurs. They are bacteria, and they are everywhere. In fact, our own body contains more bacterial cells than our own cells. Yet, we know so little about that organisms. 10 years ago, scientists knew only 5,000 bacterial species, and mostly those species who can produce beer or can cause the disease. What about all others? Traditional lab techniques to research bacteria are way too slow. Fortunately, we are living in the genomic era, which allows us to reconstruct bacterial genome from the small DNA fragments. That allows us to extend our knowledge from 5,000 bacterial species to 125 bacterial species for now. And that is my work is about. But how to study tiny organisms which you cannot see? I told you that bacteria are everywhere, which means you can grab the samples of anything. It can be soil, water, your own skin. And there definitely will be bacteria in this sample. And bacteria will contain a genome. If you manage to read these genome fragments, you can have a snapshot of all organisms present in this sample. That's amazing. But the problem is that the small fragment of DNA mixed in this sample, like sentences from different books in one big document. And your task is to separate these sentences and assemble the book of bacterial genome. You can use different parameters for that, like, for example, frequency of the letter in the sentences, or some repeated patterns in the sentences, or, for example, number of the same sequences you can find in the sample. In the end, you will have many different books, many different genomes. Some of them will belong to very well-known species, but some of them will represent completely new bacterial species. I am working with this technique in Estonian biobank, one of the biggest and well-organized biobank in Europe. 1 in 10 Estonians donated the sample to this biobank, which means you probably know somebody who participated. Using the medical data, the rich medical data from this biobank, I managed to connect bacterial species with the diseases. I reconstruct in recent 2 years more than 84,000 bacterial genomes, including 300 new bacterial species. I managed to connect 41 bacteria with the 15 diseases. I was surprised that the strongest connection for the heart failure belonged to new bacteria, which I just assembled a year before. That's why I am sure that we need to reconstruct a new species, which unlocked us the secret of our health. Thank you.